Greetings, my fellow colleagues in this PhD program. My name is Nicolaus Sándor, and I'm about to investigate the effects of micro and nanoplastics on health in my PhD. A little bit about my background. I'm sure that most of you are healthcare professionals, physicians of some sort. I'm a banker from the Central Bank of Hungary, the sole health economic analyst of the Central Bank of Hungary at the moment. Yeah, well, that's what it is. So I came with a little vision here, and this is where I imagine myself a world where environmental factors are not deteriorating human health. Since we are living in the age of epidemics, it's a relevant topic. How will that be possible? Well, the mission will accomplish the vision, which is to provide the necessary high-quality evidence to policymakers to improve the quality of life in general. My two topics are going to be, for now, two systematic reviews and meta-analysis. The first one we started in September of this year. It is investigating the effects of MNPs, or micro and nanoplastics, and health in in animals. And the second, which we started in November, is investigating the effects of MNPs and health in humans. Also a systematic review and meta-analysis. Let me talk about the first project so far. Little bit about the background. 9.2 billion tons. That is the total amount of plastics estimated to have been produced between the 1950s and 2017. It's a lot, and even more problematic it is that since 2004, half of it has been only produced since 2004. So technically, by next year, you know, everybody is going to be made out of plastic. If you think I'm joking, I am not. Here you can see on the right slide where the green little dots are. That's the human sperm. The green on it is attachment of micro and nanoplastics to the human sperm. On the right one, you can see human placenta, and the red dot there is microplastic and nanoplastics in the human placenta. So technically, right at the point where we are conceived as humans, we are also already partially plastic. And from that point on, we just become more plastic. We consume it through ingestion, inhalation, water bottles, even through air, you know, skin contact, nylon underpants, all these stuff. They are highly endocrinic disruptors. They are bad for the environment, but even worse, bad for the humans, bad for the animals, bad for everything that's living, basically. So what we're going to investigate is what the effect of MNPs on the health in vertebrates is, and how does the effect depend on properties and dose of MNPs. My PICO framework is going to be healthy non-human vertebrates, such as mice, rats, fish, who would have guessed, right? In vivo and vertebrate-driven cells and in vitro. Fish are very relevant since most of the plastic is found in the oceans, and through eating sushi and every other nice stuff, we consume a hell of a lot of it. MNPs is going to be the exposure, and the comparator will be no MNPs, of course. Outcomes will be all health and toxicity related outcomes. My hypothesis towards this study is that MNPs worsen health outcomes depending on dose and type. Size, way of entry, and chemical composition are of matter, in my opinion, but we will see. What will be the implication of this research? Well, I hope that we can create some recommendations for environmental MNP levels. I hope we can make suggestions where to focus funds and for what should be measured and how we should approach the whole issue. And more so, we could scale the burden of the social burden of MNPs. So far, my preliminary research resulted in 98 meta-analyses which we extracted on the topic. Only 3 of them focused on mammals, and the Nobel Prosper registration was found in the topic, so lucky me, I guess. Key articles-wise, firstly, we focus on the fertility of animal models, specifically here in mice, which were exposed to microplastics in laboratories. The key findings all imply that there's a huge drop in fertility, increase in inflammatory markers, decreased sperm count, decreased motility, increased deformities, And that's just fertility and mice sperm, right? So imagine what else is happening with the lungs, with the immune system, with all the other systems, not only in humans, but also in animals. My final search key can be seen here. It resulted in 13,951 articles, which were gathered from 3 databases, PubMed, AMBIS, and Cochrane Library, pretty standard. Regarding this, we finished duplicate removal, resulting in 8,527 articles, and selection will continue with my co-investigator, where is he? Szilárd Salzer. For this, I imagine the deadline to be the 21st of December, so I hope you are done with Christmas shopping, because we have a couple of days of work ahead of us, and hopefully submission will be by next spring. Let me talk about the second project, investigating the effects of micro and nanoplastics on health in humans. To me, it seems evident after the first semester of this PhD program that it's logical to start with lab animals and see it in a narrower domain, and then try to pull it together with other health-related topics of a complexer entity such as humans. What we could find so far in the background: detection of microplastics in the human penis. Sorry for bad language. Detects on the presence of human urine and kidney tissue, in the lung tissue. Then we have it in the female reproductive system. Then we have it in our arteries. Then we have it in the lung. Then we have it in the blood itself. Then it is also in other bodily fluids. So it's getting more and more. The first detection of it in the human placenta, the article was published in 2020. So what we consider is to do a single-arm study focusing on the different tissues where we find microplastics in for the framework and the clinical question. What is the prevalence and distribution of microplastics in human tissue samples, and how does this vary across different demographic factors? We would think that since it comes from seafood, a lot of it, maybe people who have access to sea are higher exposed, but that's not necessarily true. Japan has the highest plastic packaging rate in the whole world. Maybe they have more microplastics, and if they don't, why not? So these are all highly relevant questions. We chose the COCOPOP framework, which will be MMPs, tissue samples in humans. To make it brief, MMPs prevalence differ among humans' tissues and cause a multitude of health problems besides being an endocrine disruptor, is going to be the hypothesis. For the key, I'm just trying to hurry because I only have 10 seconds. Clinical or research implications. We want to show tissues which are most affected by MMPs. We want to enable to develop targeted treatment to see which should be focused on, and mapping the overall effect. Maybe MMPs affect the lung worse than the reproductive system. Spoiler alert, I don't think so, but maybe. Key article-wise, we chose the female reproductive system now for the beginning, but we, as I said, focus on other tissues too, where we can obviously see that it causes, microplastics can cross the placenta barrier, can cause deformities in the embryos, and right from the development of the child, regardless of gender, and in the next generation, can cause huge problems. So it's a hugely interesting topic with a lot of digging to be done, and I'm looking forward to put my mark on it, and with the help of this well-established, prestigious establishment, the TMK and the Semmelweis, it's going to be a huge project. So far, the topic is feasible, and that's the progress that we have made. And I would like to finalize this with a quote from Einstein. I only call it research when I don't know what I'm doing. And that's definitely the case as a finance guy. So thank you very much.
Congratulations on your wonderful presentation.
Thank you.
I'm curious, are you planning to pull together the effects of all kinds of plastics? So let's say PVC, nylon, and PET, or?
It's a very good question. I'm lucky in this regard because the most prevalent plastics is polyethylene, sorry. And So that covers around 80 to 90% of the whole cap. But on the other side, we are very interested in the different endocrine or disruptive factors of BPA or PVC or something else. That could be interesting. We have to look at the whole literature. I think mainly it will be polyethylene, but if possible, I would like to aim at making it as huge as we can and pull all the different types of plastic too.
Okay, thank you. If I may,
Please.
I might have another question or comment. I think I've read one of the articles you were referencing on detecting microplastics in sperm. And if I recall correctly, that study included like 2-digit number of participants and wasn't really representative of the population. Is this the case for the other articles where they found microplastics in other bizarre places, or were they more representative?
See, you have an article specifically you want to refer to, or?
I don't know exactly how it was or where you mentioned it, but there was a Chinese study on detecting sperm in semen.
To my knowledge, mine is made in the US, but I'm not sure.
It might be a different one, sorry.
It's fine. To answer your question, the topic itself just became popular in the recent years, like the last 2, 3, probably in the pandemic it started. Therefore, literature is all over the place, and it's not as standardized or not as well contributed to as, for example, diabetes, where we have huge or cardiac failures or something. Generally speaking, if we just think about it logically without even the literature, it's absolutely impossible to find anyone in this room who is not exposed to microplastics, and I'm sure that we can find not even micrograms, but grams in every person if we would sample the whole amount of people. So therefore, regarding this specific study, I cannot answer. Regarding the others, I can say that that will, of course, be part of the article selection, and if it's not feasible, or we put it in the forest plot, and then we can see whether it makes sense to include. But most of them have some limitations, exactly to what you refer to. We will see. I think it will be good.
All right, thank you very much.
Congratulations.
Thank you.
In your first project, as an outcome, you brought a very broad one, something like all health-related outcomes. Can you share what specific do you have in mind?
Absolutely. For example, just starting from the key articles perspective, I'm definitely looking at the sperm count of mice, the sperm motility, for example. Then we look at inflammatory markers, interleukins, TNF-alphas, and all the other sort of stuff, and maybe some outliers or specifics which we haven't found yet. But besides the standard, we are expecting some surprises to be found.
Thank you.